Hello everyone. This is part 2 of our lesson on biotechnology. And so we are still in pages 270 to 275 if you're following along with me in the Campbell Biology in Focus second edition textbook. Our focus in this second half of the biotechnology lesson is going to be on DNA fingerprinting, or a technology known as gel electrophoresis. So I guess the first point that I should make about DNA fingerprinting is that it has absolutely nothing to do with the lines and swirl pattern on the tips of your fingers. This is not about actual fingerprints, but your DNA fingerprint, which is a unique pattern that is created when your DNA is analyzed in a particular way, that pattern is unique to you as an individual, just like your actual fingerprint pattern on the tips of your fingers is unique to you. So that's why we call it a DNA fingerprint, because it is a unique pattern. The proper terminology for this, really, DNA fingerprinting is a term that's often used, but the proper term is restriction fragment analysis, and it utilizes a technique known as gel electrophoresis. Now, what this technology allows us to do is to separate fragments of DNA by size. So a sample of DNA is collected, and the DNA molecules are cut into small fragments, which can then be sorted by size. Large fragments, medium-sized fragments, small fragments, as the fragments of DNA pass through a very thick, very viscous material known as agarose gel. Agarose gel is a mixture of protein and sugar and other things that's really kind of a similar consistency to Jell-O that you might eat for a snack. So when I mention agarose gel, we're talking about literally a block of this Jell-O-like material. It's usually a rectangular block, maybe well, I guess they vary in size, but maybe 5 to 10 centimeters wide, and the length of the gel can vary greatly depending on what kind of analysis you're doing. But it's fairly thin, usually less than half a centimeter in thickness. But just imagine kind of a thick Jell-O-like material, and basically, we'll talk more about this later, but basically the samples of DNA that you're wishing to analyze are loaded into some little depressions, some little openings called wells that are at one end of the agarose gel. And electricity is used to move those DNA fragments through this gel-like material. But as you can see in this image, some of the fragments of DNA travel further than others. So some fragments of DNA don't travel very far. Those are the larger, longer fragments of DNA, whereas the tinier fragments travel further. And it allows us to separate fragments of DNA by size. Now, I'm getting a little bit ahead of myself, so let me back up here to something I mentioned here earlier, that in order to analyze DNA with gel electrophoresis, the first thing that must be done to the sample of DNA is to cut that sample, literally cut the DNA into fragments. Now, as you know, when we cut DNA in a laboratory setting, we have to use what are called restriction enzymes. We talked about this before. Restriction enzymes are naturally occurring enzymes that are found in bacteria. They are part of the bacteria cell's natural defense against virus DNA. But we use restriction enzymes in the laboratory to cut DNA and to make very precise cuts. Because remember, each restriction enzyme will only make a cut at a very specific recognition sequence. In this particular case, this is the restriction enzyme known as EchoR1. It will only cut DNA wherever it finds a GAATTC. It will ignore all other sequences of letters. But if it finds a GAATTC, which is a palindrome, I can drop down and read it backwards, GAATTC, and it's exactly the same. Only where that palindromic sequence exists on the DNA will this particular restriction enzyme make a cut. So the idea is you collect DNA samples, perhaps from 3 different individuals or 3 different species that you want to compare, and before the samples are loaded into the gel, restriction enzyme would be added to each test tube, the same kind of restriction enzyme. The DNA will be cut into fragments, and all of those fragments are then loaded into one of the wells at one end of the gel. After the samples have been cut with restriction enzyme and all the samples have been loaded into the gel is submerged in a buffer solution, a liquid that is highly conductive to electricity. This all takes place inside of a plastic box called a gel chamber. And the way this is set up, the end of the gel where the samples were loaded is near a negative electrode. At the other end of the gel is a positive electrode. And remember, this whole gel is submerged in an electrically conductive buffer solution. When the electrophoresis process is powered up, when the electricity is turned on, what happens is the DNA fragments are negatively charged. The phosphate groups on the DNA have a negative charge. So all of those fragments of DNA are going to be strongly attracted to the positive electrode, which is down at this end. And so they're going to try to make their way. They're going to literally be pulled towards that positive electrode. The problem is, to get from negative electrode to positive electrode, the fragments of DNA have to pass through this very, very thick gel. And the gel is made up of kind of a matrix of large proteins and sugar molecules. So it's difficult for the fragments to pass through. So what happens is the very shortest, smallest, tiniest of fragments have less resistance. They're able to slip more easily through this thick gel, and so they travel further, as you see here. If it's a larger fragment, it's going to tend to get stuck as it tries to pass through this thick gel, and so it's not going to travel as far. And the really, really long fragments, the large fragments of DNA, have a very tough time indeed moving through this gel, and so they don't travel far at all. And so what you have for each of our 3 samples is you have a distinct pattern that's created because the various fragments of DNA were different sizes. Now, another thing I should mention here is that there's another technology that is often used to help with DNA fingerprinting, and it really comes in handy whenever you have only a very, very small amount of DNA that you're working with. So let's say, for example, that the DNA, one of the samples of DNA that you wish to analyze, let's say you only have a very tiny amount of DNA in that sample. Maybe all you had was a single tiny speck of blood left behind by a criminal at a crime scene, or maybe a single strand of hair. So there's going to be cells in that. There's going to be DNA, but it's a small amount of DNA. And it's such a small amount of DNA that it might be impossible to actually see visible fragments on the gel. And so a very handy technology that every biotech lab has in its arsenal is a machine called a polymerase chain reaction machine, or PCR. The purpose of PCR is to amplify DNA samples. That is, to make many, many, many copies of a sample of DNA. This is done completely in vitro. So in other words, this is done outside of the cell. This is, you've got blood, or you've got hair or skin or whatever, and you've already extracted the DNA from the cells. You then place that extracted DNA into this machine, and it makes copies or amplifies the DNA so that you have a larger amount of that DNA to study. So one of the purposes for that is to make it possible to analyze DNA even when you've only got an extremely small amount of that DNA from a crime scene, from a fossil, whatever it is that you're trying to study. So I want to talk just briefly about how PCR works. This is kind of the inner goings on of a PCR machine. And you can see there are various chemicals that are inside the machine. There's DNA polymerase enzyme. There are small pieces of RNA primer that can be inserted into the machine. And then there are lots and lots of nucleotides. So just don't be confused by these abbreviations. This would be adenine, cytosine, guanine, and thymine. And so what you're looking at right here, where it says DNA targeted sequence, this would be the small amount of DNA that I'm loading into the machine that I actually wish to copy. And if we're going to copy that double-stranded DNA, as you know, the first thing we need to do is to separate the two strands of DNA. Inside a PCR machine, this separation of DNA strands is accomplished by heat. The machine simply heats up, and at a certain temperature, all of the hydrogen bonds in between the base pairs will temporarily break, and the two strands of DNA will separate. Then, small single-stranded pieces of RNA primer will attach to the DNA strands that are to be copied. Now, this is kind of an important step here, because sometimes with PCR, you're interested in copying the entire genome. Maybe we've got DNA from a crime scene, and we want to make copies of the entire sample of DNA. Well, you can do it that way, but perhaps you're interested in only copying a very small portion of the DNA where a particular gene is located. Maybe you're interested in studying the cystic fibrosis gene, and so you want to only copy the small area of DNA where you know that cystic fibrosis gene is located. So how would you do that? Well, this PCR copying technology is extremely precise. You can design these little single-stranded primers so that they will attach to exactly the location on the DNA where you want to copy. So the primers attach, and remember, the purpose of primer is that the enzyme DNA polymerase will attach to the 3' end, and it will begin building a copy strand, a complementary strand right here that is complementary to the original strand. And on the other side, DNA polymerase will also attach to the 3' end of this primer and build a complementary strand. And so when this process is complete, look at what we have. We now have two DNA molecules, and they are both an exact replica of the original DNA molecule. Then, after these three steps have been completed, the machine simply heats back up. It goes through another heat cycle. So both of these DNA molecules are unzipped or separated and copied. And so when we're finished with cycle 2, we now have 4 copies of this DNA. The machine heats up again. We go through another cycle. Now we're up to 8 copies. And again, 16 copies, 32 copies, 64 copies. So with each new cycle of PCR, we double the number of copies of that DNA, either the whole DNA sample or maybe just the one area of the DNA that we want to study. Like I said, you can design these primers to attach wherever you wish for them to attach, and they will the DNA will only be copied in the place where the primers have attached. Now, one thing may have occurred to you as I was explaining this. This whole mechanism, this whole machine, the PCR machine, is completely dependent upon an enzyme, an actual biological enzyme called DNA polymerase, because that's the enzyme that actually makes the copies of the DNA. Well, enzymes don't like to get hot. Enzymes, remember, when they get hot, they denature, they unfold, they lose their functionality. And you're talking about a biological enzyme that has to function inside a machine that gets pretty darn hot. It goes through heat cycle after heat cycle after heat cycle. So the way around that very real problem is to not use human DNA polymerase, not use plant DNA polymerase, but to use DNA polymerase from a very specific source. The DNA polymerase that is most often used in a PCR machine is called Taq polymerase. Taq stands for a species of bacteria, Thermus aquaticus, and this is a bacteria that is very thermophilic. It lives in hot springs where geothermal vents are releasing lots of hot gases from the Earth's mantle. This bacteria can survive in temperatures as high as 176 degrees Fahrenheit, which is a temperature that would kill most bacteria. But this bacteria thrives in that temperature, and it has an unusually heat-stable DNA polymerase enzyme. In other words, heat will not damage this DNA polymerase because it comes from a heat-loving species of bacteria. And so that's the key to making the PCR machine work. Taq polymerase from Thermus aquaticus. By the way, this is a little off topic, but today's extra credit secret code word is Jennifer Doudna. Jennifer Doudna. Dr. Doudna is a well-known biochemist and molecular biologist, and if you're not sure how to spell her name, look it up. Read a little bit about Dr. Jennifer Doudna. Fascinating individual, and we're going to be learning more about her soon. Okay, so let's talk about the basic premise of DNA fingerprinting. One of the fascinating things that has come out of recent genome research is that there's an astonishing amount of similarity between the DNA molecules, the nucleotide sequences of different species. 50% of your DNA, roughly 50%, is identical to a banana, which is kind of bizarre when you think about it. If you compare your DNA to another human, a person from Africa or Europe, or Mongolia or anywhere else on Earth, you would find that 99.9%, at least that much, of your DNA is identical to every other human on Earth. So if that's the case, if my DNA is so incredibly similar to yours, how is it that our DNA fingerprints, this pattern of dashes that's created by electrophoresis, how can that pattern be so different? So let's look at two different individuals. I just made up some names here, Abe and Bob. We're looking at Abe's DNA and we're looking at Bob's DNA, and we know that they're going to be extremely similar because Abe and Bob are both members of the same species, Homo sapiens. But even a very slight difference, only one base pair is different between Abe and Bob in this particular part of the DNA that we're looking at. Abe has a CG base pair, and then Bob has an AT in that same spot. But notice how their DNA fingerprints look really, really different, and here's why. Remember, we're going to cut the DNA samples with a restriction enzyme. And we're going to use the same restriction enzyme to cut both Abe's DNA and Bob's. Now, this particular restriction enzyme only recognizes CCGG. So it makes a cut between the two C's, zigzag, and then the two C's here. So it makes a zigzag cut right here. And then if it finds another CCGG, there's another one, it makes a cut here. Well, when we cut Bob's DNA, we're only going to cut here because he doesn't have a CCGG in this location. So the result is when we cut Abe's DNA and Bob's DNA with the same restriction enzyme, we get a different number of DNA fragments. Notice Abe now has 3 fragments after we cut his DNA twice. We have 3 fragments of DNA. Bob only has 2. And look at the size difference. Bob has this enormous fragment of DNA that goes from here all the way to this cut mark. That's a long piece of DNA. So when we load that in the gel right here at the negative end and we turn on the electricity, that Z fragment is not going to travel very far because it's really, really large. The Y fragments are small, so they'll travel further. And remember, Abe has this fragment here, which is kind of a medium-sized fragment. So it'll travel further than, say, the X fragment, but not as far as the Y fragment. The point is, Abe and Bob have 2 big differences. They're going to end up with not only a different number of fragments, but the lengths of those fragments are going to vary as well. Now, one thing that I should go back and mention is when you load samples of DNA into the gel, that's a very important step, because if you don't successfully get the sample of cut DNA fragments into that little tiny well, then you're never going to see anything on your gel. And so mixed with the DNA and the restriction enzyme, another thing that's added to that sample test tube is something called loading dye. DNA is colorless, so you can't see DNA when you load it into a gel. So one of the things that's mixed with the DNA sample is loading dye. It's usually a bluish color or sometimes an orange colored dye that is mixed with the sample. And the purpose of loading dye is twofold. Let me jump over here. The functions of loading dye, first of all, are so that you can actually visually confirm that you have successfully loaded a sample of DNA into a well. You can actually see that well fill up with the orange color or the blue color, whatever color of loading dye. And then you'll know I have successfully loaded the sample. The other purpose of loading dye is the loading dye will actually move through the gel faster than any of these DNA fragments. And so you can't quite tell this in the picture, but the reality is you can't see any of these fragments of DNA. They're completely colorless. You actually there's a step at the end where you have to stain the entire gel to make the DNA fragments become visible. So when you're running the gel, the only thing you can actually see moving through this gel is the loading die. And basically you want to run the gel until the loading die reaches almost the end of the gel, but not quite. If you let the gel, if you leave the electricity on too long, then all of your fragments of DNA will run all the way off the end of the gel and you won't see anything at all. So loading die helps us determine when it is time to turn off the power supply, because the loading die is going to move through the gel slightly faster than the actual DNA fragments. If we let the loading die run to the end of the gel, we know that all of our fragments are going to be somewhere back behind that loading die, which is where we want them to be. Okay, earlier I showed you a simple example with Abe and Bob and DNA samples that we collected from those two guys. This is a similar type of example, but it shows a little more detail. So this time we've got Ned and Ted. Let's assume that these two guys are unrelated, but like any two humans on the face of the earth, their actual DNA nucleotide sequences are going to be very, very, very similar, 99.9% similar, or 99.9% identical, actually. And so when we analyze DNA with electrophoresis, we're really looking at very, very slight differences in the DNA. So let's assume that we have collected a DNA sample from Ned. We'll call that sample 1. We have also collected a DNA sample from Ted. We'll call that sample 2. And we have put those two samples into two separate test tubes. And we have added a restriction enzyme to the DNA. We're going to use the restriction enzyme EchoR1. That enzyme will scan the entire nucleotide sequence, and it is searching for the recognition site GAA-TTC. Whenever it finds GAA-TTC, it will make a zigzaggy cut through the DNA. And then it will keep scanning. Here's another GAA-TTC. There's a cut. So Ned's DNA will be cut twice, producing 1, 2, 3 fragments. Now we call these fragments of different lengths. Get ready for a great big word: restriction fragment length polymorphisms. RFLPs. What does that mean? It means that each fragment of DNA has a slightly different length. The A fragment is 1, 2, 3, 4, 5, 6, 7, 8 base pairs long. Fragment B is a little bit longer, 1, 2, 3, 4, 5, 6, 7, 8, 9 bases long. And fragment C is the largest of the three, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, I think 13 if I counted right. So they are different lengths. Now remember, we're taking Ted's sample, which is in a separate test tube, and we're also cutting his DNA with the exact same EchoR1 restriction enzyme. Here's a GAA-TTC, so we're going to make a cut here. But notice right here on Ted's DNA, it's GAA-TC. That's not a recognition site. There's a slight difference between Ted's DNA and Ned's DNA. There's the difference right there. So GAA-TC is not a cutting site. And there's not any more on this particular stretch of DNA. So Ted's DNA is only going to be cut into 2 fragments instead of 3. Fragment A and fragment D. Notice how long fragment D is. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. 22 bases long, way longer than even the largest fragment from Ned's DNA. We're also going to mix into these two separate test tubes loading dye. So we're going to mix some red loading dye in with Ned's sample, and we're going to mix some blue loading dye in with Ted's sample. Notice both samples are loaded at one end of a rectangular block of agarose gel, and we're going to place that into the gel chamber right beside the negative electrode. This entire gel will be submerged in an electrically conductive buffer solution, and when the electricity is turned on, the negatively charged fragments of DNA become strongly attracted to this positive electrode, and they start migrating towards the positive electrode, moving as fast and as far as they can move. But remember, the fragments are all different lengths. So fragment A, which both men have, is a really tiny fragment. So it's able to slip through the thick gel, and it travels a fairly great distance. Fragment B is kind of a medium-sized fragment, so it doesn't travel quite as far. Fragment C is a large fragment, so it travels even less distance. And then look at Ted's DNA. Remember, he has this huge fragment D, which is really, really long. And so that's not going to travel very far at all. And so when we look at the DNA fingerprint, that's the pattern that's created, two things are different. The number of fragments is different, and the relative size of those fragments is different. And that's why we get this unique pattern. It's completely 100% unique to you. Your DNA fingerprint is unlike anybody else's on the face of this earth, unless you happen to have an identical twin. Then you would have virtually the same DNA fingerprint. One other thing I'll mention here, I've mentioned it briefly before. At the end of electrophoresis, when this gel is removed from the machine, these fragments of DNA are completely invisible. They're the same color as the gel. You cannot see them at all. So there's a couple of steps that take place at the end. The entire gel is stained with either some sort of dark stain. We usually use methylene blue in the laboratory here at Union Pines. You can also use dyes that will glow under ultraviolet light. So there's different ways to do it, but the gel has to be stained and then destained so that the fragments or the bands of DNA will show up against the background of the gel. And that's something that I typically go into more detail about when we actually do these labs in class. Now, speaking of labs, this is the electrophoresis experiment that I do normally each year with my AP biology students. Each lab group is given some melted agarose, and so they have to pour their own agarose into a rectangular mold to create the gel block here. And they have several different wells at the negative end of the gel. And each lab group is given 6 samples of DNA that have already been cut with restriction enzyme. So samples A, B, and C are DNA samples from 3 different children. They're all siblings. Sample E is DNA that has been collected from the father of these 3 children. And this father has colon cancer, and he has a known mutation that predisposed him or made him more likely to develop that colon cancer. And so the DNA samples here, the PCR that was set up to create these samples did not copy the entire genome. It only copied the portion of DNA where this particular cancer mutation may be located. So what we're looking for when we look at the DNA fingerprint in this lane, the E lane, we're going to look at that pattern and we're going to compare it to the patterns of each of the three kids. And what we're hoping is that none of those three children inherited that cancer predisposition mutation from their father. We also have a sample of wild-type DNA. Sample D is taken from an unrelated individual who does not have that particular cancer mutation. And we're hoping that the three kids all will have inherited a normal wild-type allele and not the cancer-causing mutation that their father has. And then there's one other added mystery. Two of these three kids are identical twins. And so the lab group has to figure out which two kids are identical twins. That's easy to figure out because two of those kids will have exactly the same DNA fingerprint. And then finally, there's an F sample where no source DNA was placed into the PCR machine. And so that should be a blank lane with no DNA fingerprint. The purpose there is just to identify any contaminants. If you end up with any DNA fragments in the F lane, that means somehow you contaminated the electrophoresis process. So it's really just these first 5 samples that we're interested in analyzing. It's a really fascinating experiment that I enjoy doing with my AP biology students each year. Our next lesson, this seems like a bit of a jump, but it is related to the expression of genes and the regulation of gene expression. We're going to be looking at cell specialization and also the related topic of embryology. So to prepare for that next lesson, you need to read page 322, just that one page in Biology in Focus, and then skip over and read pages 764 to 767. I hope you're all doing well. I miss you, and I hope to see you soon.